Here's a bone. That's a great picture. That is a bone. Maybe that's your femur or whatever. It's a regular old bone. If you were to saw that bone in half on a bandsaw like this, you're going to see that there's two distinct regions to it. You're going to notice that there's this big hollow cavity in the center. There's actually yellow marrow in there, yellow bone marrow. And the sort of tubular portion of the bone is called the diaphysis of the bone. Over here and over here at the ends, you're going to have what literally looks spongy. It's called spongy bone. I'll bring in a real bone and you can see this. My dog likes to chew on the frozen bones that I get from Adam, so I'll bring a piece in. I'll bring my dog in, so I keep saying I'm going to do that. Anyway, you got this spongy bone there, and the spongy bone is where red bone marrow is. So the red bone marrow and the yellow bone marrow are very different. Yellow bone marrow is just this fatty material. It's really mainly there just to store calories. There's a lot of fatty acids in there. The red marrow is where you get blood cell formation to happen, at least in an adult human. The process of hemopoiesis happens here. Hemo means blood. This means to form a cell. So hemopoiesis is blood cell formation. If you want to be specific and say red blood cell formation, you can call it erythropoiesis. So that's going to happen in adult humans at red bone marrow. That's where you find red bone marrow. At the ends are what are correctly called the epiphyses of the bone. So this is an epiphysis of the bone. Here's the other epiphysis. The plural is epiphyses, with an ES at the end. And so that's kind of the anatomy of an adult bone. That's where the red marrow is located. That's where the cells that give rise to blood cells live. Actually, white blood cells come from that red bone marrow as well. We'll talk about that in a second. But there are stem cells. There are cells that are semi-specific. They're not totally, completely differentiated. They haven't completely decided what they want to be when they grow up, that live in the red bone marrow. When you influence them with the right chemicals, including some hormones, you're going to get them to turn into the blood cell that you need. For example, if you want to get one of these stem cells to turn into a red blood cell, you hit it with the right chemicals, and it turns into a red blood cell. As it's doing so, it's kind of a step-by-step process, there's a precursor to that red blood cell that's living in here that pretty much has all the equipment of a red blood cell, but also has a nucleus in it. And then on its way out the door into, say, here's a capillary. Before it leaves here and gets into the circulatory system, it tosses its nucleus out and enters, and that's how you get a red blood cell.
So how does it know what to do without a nucleus?
Well, it's limited. It can't reproduce now, right? It can't do mitosis because it doesn't have a nucleus. So its main job is just carry a bunch of oxygen around and help metabolize and allow for the transport of CO2. And it can do that because it's packed with hemoglobin and it's packed with carbonic anhydrase. So it's able to do that. But what you're suggesting is, yeah, but it can't sustain that, right? Because it can't make new hemoglobin, it can't make new, because it doesn't have the DNA to code for it. And that's true, which is why they don't live very long. They live about 120 days. So you're constantly making new ones. Because the old ones, you know, they got enough to go for a while and then they don't work anymore, they die, they get processed out, their parts get reclaimed and repurposed. Your liver and your spleen help to do those jobs, by the way. And you always make new ones. Yeah. Make sense? Okay, yes?
For the nucleus that's kind of ejected out before the red blood cell enters the circulatory system,
right.
Is that also kind of just recycled?
Yeah, all those materials will be, the body's pretty conservative about that. If it's something that can be salvaged, like nucleotides, chop them up and reuse them, rephosphorylate them, and yeah. Even the dead red blood cells, even the hemoglobin in the dead red blood cells, because there's protein there and there's iron there, eventually the dying red blood cells will wind up at the liver, and the liver will extract the iron from it and turn the hemoglobin into what's called hemosiderin, which is a yellowish material, and that becomes part of your bile that goes to your gallbladder, that goes to your intestine, that eventually gets excreted from you. If you have jaundice, in other words, if you have a compromised ability to do that, you're going to have a bunch of that material, that yellowish stuff building up in your body, and that's going to give you the yellow pallor that one has when they have jaundice. It's actually processed hemoglobin that used to be a red color, but when you turn it into this other material, it has more of like an amber color. And that's that. Yes?
Was diaphysis? I think I missed it.
Diaphysis,
Yeah.
it's the tubular portion of the bone.
And
Epiphysis, it's the end of the bone,
Oh,
a
that's
circle.
okay.
Yeah, it's the parts of the bone. I mentioned there's a bunch of chemicals that influence all this. There are many, but I'm going to mention one, okay? Because it's a hormone, and we talk about hormones, we should throw it in here. Let's say you, for whatever reason, you're not carrying as much oxygen around in your blood as you should be. That might mean that you don't have enough red blood cells for whatever reason. You need to make them faster. It might mean that there's just not enough oxygen in your environment. You know, maybe I moved to Denver and I'm living 6,000 feet above sea level. There's less oxygen in the air. It's going to take me a while to adjust to that. It might mean, I don't know, maybe I just donated a pint of blood and I don't have as many blood cells as I used to have. No matter what the reason, a decreased oxygen concentration in my blood is a big red flag to your body. You say, well, we better make more red blood cells to help compensate for our compromised ability to transport oxygen. So let's take that situation and see how you compensate. So I have a decreased partial pressure of O2 in the blood. Partial pressure, right. We have said this before, I'll throw it out there again. If you want to know what's going on in the blood, what organs should you ask? Kidneys.
Yes.
They're getting 20% of your cardiac output. We saw that with things like the renin-angiotensin aldosterone action. They're small, but they get a lot of cardiac output. So a good place to sort of get a feel for what's happening in the blood is at a kidney. The kidneys notice this first. So, detected by kidneys. And in response, they release a hormone. It's called EPO. EPO is an abbreviation, obviously. It stands for this. Remember a minute ago I said erythropoiesis is red blood cell formation? Well, the hormone erythropoietin stimulates red blood cell formation. So the more EPO you dump into the body, it's a hormone, it's a peptide hormone. The more EPO you dump into the body, the faster you're going to make red blood cells. That bone marrow is going to crank out red blood cells faster than it did when there was less EPO. So it's a supply and demand. Hey, not enough oxygen around? That's probably could be compensated for if we got more red blood cells. Let's make more red blood cells faster. How do you make that happen? Release more EPO. One of the ways, there's more than one way to do this, but one of the ways people blood dope is just by using recombinant EPO. It's a peptide hormone. It's very easy to produce recombinantly and buy on the black market. And if you take EPO, you will have more red blood cells in your body than if you didn't take EPO. So that's one of the forms of it's outlawed by every athletic organization there is, but it's one way to do it. It's even arguable as to how much it's going to help you in athletic performance. It depends on what kind of, if you're a duration athlete or if you're running a marathon versus if you're a weightlifter, it's probably two different effects. Anyway, yeah, blood cell formation. Good. All right. Leukocytes. They're the white blood cells, but there's more than one kind of white blood cell. So we're going to have to break down Roman numeral 2 into A and B and C, etc. So let's do this. I'm going to give you categories of white blood cells and then name a couple examples in each category. You can take, like I said, there's a whole variety of different white blood cells floating around in your body. You could take a collection of them. You know, you take a drop of blood, you gather all of them together somehow, or you photograph them all under the microscope, and then you say, okay, what kinds are we looking at? How is each one different from the other? You would first be able to say there's two major categories. There are what are called granular white blood cells, and there's agranular, or not granular white blood cells. Some people just call them granulocytes and agranulocytes. Called granular because when you look at them under the microscope, there's what look like big grains of stuff in the cytoplasm. If I look at a cell like this, and they're more or less round like this. If I look at a cell like that, and I see a nucleus, and then I see these, you know, that's a granulocyte. If I look at it under the microscope and I see a nucleus and I don't see that, that's an agranulocyte. It's pretty straightforward. They're easy to see. We'll look at them in lab next week. Some examples of granular versus agranular. If you go get a physical, so you get an annual physical, one of the things they're going to do in your blood work is tell you how many granular, agranular cells, how many of each kind, what's the percentage of these versus those in your blood, because having too many or too few of a certain kind would be indicative of a certain disease or disorder. Depends on which one. So examples of granulocytes that you will find in your normal peripheral whole blood would be things like, let's start with eosinophils. There's no particular reason to why I did them in that order, I guess. So, eosinophils, basophils, and neutrophils. You know what eosin is. What is eosin? The pink staining dye when you stain tissues to look at under the microscope, right? So it's pinkish, reddish in color. Philios means what in Greek? Like something that is hydrophilic. Loves water. Philios means to love in Greek. Hydrophobic means to fear. That's the opposite. So eosinophils love eosin. When you stain an eosinophil with H&E, remember H&E? The granules are going to prefer the pink eosin over the blue hematoxylin. So it's going to look kind of like this. The granules are going to look more towards the reddish side, the pinkish reddish color. Basophils. Hematoxylin, that's the blue dye in the H&E combination that we talked about a while ago. Hematoxylin is a basic substance. It's, you know, if you look at acids and bases, it's a base. So if this is a basophil, the granules are going to prefer the blue, which happens to be basic, over the pink. So the granules are going to look blue. That's one way to tell that this is a basophil. There's other ways too, but that's one way to tell it's a basophil. The granules look more shifted towards the bluish color. I mean, I guess you could have called them hematoxylinophils, but that's too hard to say. So basophils is easier because hematoxylin's basic. Let's call them a basophil. It loves the basic dye. Guess what the neutrophils like? Both. They're neutral. They like both the acidic and the eosin is acidic. They like both this stuff and this stuff. So guess what color those granules are going to be? If you get red and blue together, I know you know that from kindergarten when you mix paint. Purple, yes, exactly. These aren't trick questions. They're really easy. So they're going to be more towards the purplish color. There are other ways to tell which one you can look at the size and shape of the nucleus. We will point this out when I actually have the real ones up on the screen instead of my terrible picture. But if you scan your average everyday healthy person's blood, the most numerous of those three by far is going to be the neutrophil. 70, 80%, something like that off the top of my head is going to be neutrophils. Neutrophils are responsible for a whole bunch of jobs in the body, including killing bacteria. That's one of the things they are good at. We're going to look at immune response next week, and we'll consider the role of a neutrophil and some of the other cells that we'll deal with today. But they're good at killing off bacteria. The basophils and the eosinophils also have defensive jobs. They also are part of your immune system, but they can be a little more specific. For example, eosinophils are not very numerous, maybe like 1 to 2%, or basically a half to 2% of these are eosinophils. You got to look around to find them. There's not that many of them there. They're particularly good at killing parasites rather than bacteria. So if you have a parasitic infection, you're going to have eosinophilia. You're going to have a whole bunch of eosinophils. That's one of the ways to diagnose a parasitic infection is by saying, oh my God, you've got a lot of eosinophils here. You've probably got a parasite. They also have some role in allergy as well. The basophils also play a role in allergic responses. So in other words, each of them has a specific job. Those granules, they're all different things. They're mediators of the immune response that we'll look at next week. They're enzymes. They're packets of material that the cell needs to do its defensive job. So there's your granulocytes. There are subtypes of each of these. For example, next week we'll look at a mast cell, which is a subtype of one of these. But there are your three basics. If you get your blood work paperwork back from your physical, they're going to say glucose, and then underneath it it'll say eos, basos, neutros, and it's telling you what percentage of each, it's saying, is this within normal range or should you worry about something? But then there's a couple of kinds of agranular cells. By the way, they also test for when you do your physical. These would include, which order should I do these in? Let's do monocytes first. Monocytes and lymphocytes. You look at these under the microscope, the cytoplasm is not green, hence they are agranular. The nucleus tends to be really big in these cells. That's another way you can recognize them. You'll see that next week under the microscope. A monocyte, here's one function of a monocyte. It relates to something you already know. Remember we talked about macrophages, alveolar macrophages? I said, oh yeah, they used to be white blood cells, and then they crawled through a capillary wall and started crawling around solid tissue. Now we call them macrophages. Well, guess what cell they used to be? They used to be monocytes. So monocytes give rise to macrophages, and if you recall, macrophages don't just exist in the lung. They exist elsewhere in the body. There's macrophages crawling around in your brain and in your liver and in your skin and other solid tissues just to do surveillance. Kill some bacteria if they show up, clean up the mess when there's a dead cell laying around, things like that. So where did they come from? They used to be circulating monocytes that squeezed their way through a capillary wall and got into solid tissue. Lymphocytes. There's two major subtypes, and then we will list sub-subtypes next week. The two major subtypes of lymphocytes are, how do you want to do this? I guess I'll do lowercase a and lowercase b. How's that? I'm really breaking this down. B lymphocytes or B cells, and T lymphocytes or T cells. Each of them is important in the immune response, and we're going to outline the immune response next week. So they're coming up again. We're going to talk about them next week and be more specific. And I'll tell you, B cells make antibodies, and T cells are capable of doing a bunch of things. And so for the moment, let me just put them on the list. And we'll come back to them. Okay. So that's a quick run through white blood cells. I only have 2 minutes left, so I'm going to be really quick about thrombocytes. Thrombo means clot. Now, thrombocytes are commonly called platelets. They're not actually cells. They are formed elements. They are structures that were very specifically built by the body, but they're technically not living cells. They're sort of the parts of chopped up previously living cells. So if you go into your red bone marrow, One of the cell types that you're going to see floating around in your red bone marrow is a cell type called a megakaryocyte. Cyto means cell. What does mega mean? Big. What does karyo mean? Prokaryote, eukaryote. Nucleus. These are cells with a big nucleus. Okay. Megakaryocytes. They exist in the red bone marrow, and when they undergo an appropriate set of events, which include an apoptotic set of events, they systematically carve themselves up into pieces, not just useless little fragments, but pieces that can actually have a function, pieces that have, say, certain proteins and enzymes in them. Those pieces technically aren't living cells, but they do enter the bloodstream, and they do have a function. They're called thrombocytes. Again, we commonly call them platelets. They're much smaller than the cells, but they're formed in a particular way. That's why we call them formed elements. And since we don't have any time today, I'll tell you all about their function on Wednesday, and we'll talk about how to stop blood from flowing and platelets and blood clotting and all that good stuff on Wednesday. And we'll talk about inflammation on Wednesday too. Yay.